Central dogma is the process of transcription and translation. In this video, we will demonstrate how DNA turns into RNA, which then turns into proteins. Transcription is the synthesis of an RNA copy of a segment of DNA. RNA is synthesized by the enzyme RNA polymerase. Replication. DNA is a molecule made up of two strands that are twisted around one another to create a double helix. DNA is made up of four chemical bases, which are A, T, G, and C. The two strands are complementary to each other, and they have a 5' and 3' end. They also run in opposite directions. This determines how each strand is copied. One strand is called the leading, and the other is called the lagging strand. The first step in DNA replication is the unzipping process. This is when the two strands separate due to an enzyme called helicase. This then results in the creation of the replication fork. The separated strands now provide a template for creating a new DNA strand. An enzyme referred to as MACE is responsible for binding this process. This enzyme makes a small piece of RNA called a primer, which is a starting point for the construction of the DNA strand. RNA polymerase then binds to this primer and makes a new strand. RNA polymerase can only add DNA bases from the 5' end to the 3' end. The leading strand is made continuously, meaning that DNA polymerase is being added to each end from 5 to 3. However, the lagging strand cannot be built this way. It runs from the 3' end to the 5' end. DNA is made into small sections called Okazaki fragments. Okazaki fragments bind with an RNA primer. After this, RNA bases are added. Further down the lagging strand, the next primer is added and another fragment is built. These steps will then be repeated. When RNA has been made, all of the RNA primers are removed from both strands and another RNA polymerase fills the gaps with DNA. Then the ligase then seals the fragments to form a constant double strand. Replication is considered semi-conservative because each DNA molecule is made up of an old and a new strand.
Transcription is the first step of gene expression. During this process, the DNA sequence of a gene is copied into RNA. There are different types of RNA, each encoded by its own type of gene and all provide a different function. mRNA, or messenger RNA, encodes amino acid sequence of polypeptides. tRNA, or transfer RNA, brings amino acids to the ribosome during translation. rRNA, or ribosomal RNA, makes up the ribosome and plays a role in protein synthesis. DNA and RNA both have 4 nitrogen bases. DNA has adenine, thymine, cytosine, and guanine. RNA has adenine, uracil, cytosine, and guanine. DNA is double-stranded while RNA is single-stranded, which is why DNA can't leave the nucleus, but RNA can.
Translation. Translation is also known as protein synthesis. Translation has 3 stages: initiation, elongation, and termination. Messenger RNA is a substrate for translation. mRNA contains codons that will encode for specific amino acids. tRNA contains an anticodon that is complementary to an mRNA codon. For example, here we have AUG. A complementary base would be UAC. AUG is usually the main start codon here. Here you see tRNA, and at the top you also see an amino acid. The amino acid corresponds to the codon. The amino acid is MET. Initiation is when tRNA enters through the A site. As it moves along the ribosome, acid breaks off and sits in the protein exit channel. The tRNA then exits from the E site to be recycled. This process is then repeated, resulting in elongation. As elongation is at play, the growing polypeptide chain is continuously growing and being attached by peptide bonds. When a stop codon enters the ribosome, a release factor enters the A site. Translation is now terminated and the polypeptide now exits and becomes a protein.